All right, guys, we are going to do our final section here, which is on meiosis. Again, you'll just be paying attention to the portions in red, and I actually have those highlighted on the slides. So you're not going to see it in my video, but if you go to the other link that's in this document, you'll click on that and that'll show you the red portions. So we're looking at the formation of haploid cells, and that is what meiosis is. It's how we produce our sex cells or our gametes. Human gametes are the sperm cells, which come from the men, and the egg cells, which are females. And you can see the picture here below. There's a sperm, there's an egg. Eggs are considerably larger than the sperm. Each gamete is 1N or haploid, so it has 23 chromosomes. The fusion of egg and sperm is going to create a zygote that is 2N or diploid, and it has 46 chromosomes. So there we go. Now, whenever they combine, they're going to form a blastula, which is going to divide for about a week, and then it'll implant in the uterus and actually start to become a baby. Cells undergoing meiosis go through the G1S and G2 phases, just like they did in mitosis. So the DNA will be replicating, and it is going to go through 2 divisions instead of just 1, like it did in mitosis, and that's how we end up with the haploid cells. So the first cell division is called meiosis 1, the second is called meiosis 2. So you can just take a look at that diagram here. Interphase, this is where everything's copied. We get our daughter nuclei, which would be the mitosis version of it, and mitosis would stop right here. But because we want to have haploid cells, we have to cut that number in half. So we're going to divide again and get 4 cells. Meiosis 1 is very similar to the stages of mitosis that we went over in the last section. However, since the cell is dividing twice, we give numerical names to each phase. So prophase of mitosis is called prophase 1 in meiosis 1. Metaphase of mitosis is called metaphase 1 in meiosis 1, and so on. So we'll start with prophase 1. The DNA is going to coil tightly into chromosomes and the spindle fibers will appear. The nuclear membrane and nucleolus will disassemble and disappear. Synapsis is the pairing of homologous chromosomes in meiosis. So in prophase 1, homologous chromosomes are going to pair together. Remember that one chromosome is going to be from the mom, one chromosome is going to be from the dad. So here's a karyotype. This would be for a male. One of the chromosomes would be from mom. One of the chromosomes is going to be from the dad. A tetrad is a pair of homologous chromosomes. So if I have the paternal set here, maternal sets here, when I pair the paternal and maternal together, we're going to get a tetrad. Crossing over is a process where portions of the chromatids on homologous chromosomes are going to break off and get exchanged with portions of the chromatids on adjacent chromosomes. This means that some of the DNA from the mother chromosome gets switched with DNA from the father chromosome. So there you can see what's happening as far as the switching over goes. Just a leg of the chromosome is going to kind of connect, and then you're going to end up with some mom DNA in the dad chromosome, some dad DNA in the mom chromosome. Genetic recombination is the production of offspring with combinations of traits that are different from that found in the parent. So this is why you don't look identical to either one of your parents. You may have some of their similar traits like hair color, but you don't have every single trait. And I know some people say, well, I look like a spitting image of my mom, or I look like a spitting image of my dad. And that's true in some sense of the word, you do, but there are some slight differences as well. Metaphase 1, we're going to have the tetrads line up at the midline of the dividing cell. Spindle fibers are going to attach to the centromeres of one of the chromosomes in the homologous pair. In anaphase 1, each homologous chromosome is going to move to the opposite pole of the dividing cell, and this is going to lead to independent assortment. Independent assortment is the random separation of homologous chromosomes during anaphase that results in genetic variation. So there you can see the crossing over. And here we have it again. These are different combinations that could happen. You could end up with a cell getting mom and dad DNA, or a cell getting some dad and mom DNA. Maybe the cell is just going to have mom DNA. Maybe the cell is just going to have dad DNA. It depends on how the cell separates. Telophase 1 and cytokinesis 1. So the chromosomes are going to reach opposite ends of the cell. The cytoplasm is divided between the two newly formed cells, and each of our two new cells has a haploid number of chromosomes. On meiosis 2, which is immediately going to follow meiosis 1, so they really don't take a break, we're not going to be copying the DNA this time. We're strictly here to divide. So we're going right back into it. Prophase 2, the spindle fibers are going to appear. The nuclear membrane and nucleolus will disappear. No crossing over is going to occur this time around. In metaphase 2, the chromosomes will be lining up in the midline of the dividing cell with the chromatids facing opposite poles. Spindle fibers are going to attach to the centromeres of those chromosomes. In anaphase 2, the chromatids are going to separate and move toward opposite poles of the cell. And in telophase 2 and cytokinesis 2, a nuclear membrane is going to form around the chromatids of each of the four new cells. The cytoplasm is divided between the four new cells, and each cell is haploid and contains half of the original cell's DNA. Now, in animals, the only cells that divide by meiosis are going to be those that produce gametes. So your autosomes do not go through meiosis. We give specific names for these cells that are going through meiosis. Spermatogenesis is what the male cells go through. And as it goes through that, you're going to produce 4 viable sperm cells from 1 sperm cell. Oogenesis is the production of female gametes known as egg cells or ova. During cytokinesis 1 and cytokinesis 2 of oogenesis, the cytoplasm of the original cell is divided unequally between the 4 new cells. This leaves 1 large mature egg cell and 3 smaller products known as polar bodies that will eventually degenerate. So here we have our 1 egg cell, and by the end of it, we only get 1 egg. These other ones here will not be eggs. So guys get 4 sperm cells, we get 1 egg cell. Sexual reproduction is the production of offspring through meiosis and the union of a sperm and an egg. Offspring produced by sexual reproduction are genetically different from the parents due to crossing over and independent assortment in meiosis. And that is where I will leave you as far as our notes are concerned. Make sure you take a look at the slide portion of it, because that is where you'll find the highlighted sections. And then you will be turning in your notes into the bin right after this. Alrighty guys, I will talk to you later.